Hi, my name is Flora, and today I'll be talking to you about the effect of vitamin E on learning and memory in chronically stressed rats. The way that I want you to have a think about this is imagine learning and memory in the core of what we're talking about, which is really important because learning and memory forms the basis of everything that we do, right? However, there are some things that can actually impair learning and memory. For example, chronic stress. Chronic stress changes our normal brain structure, including areas of the brain such as hippocampus, amygdala, and the prefrontal cortex. Animal models have also shown that chronic stress can impair hippocampus-dependent learning and memory. So here's learning and memory. Chronic stress comes, impairs it. There is also something called the brain-derived neurotrophic factor, also known as BDNF. And BDNF is a neurotrophin involved in neuroplasticity, including learning and memory. What's really, really interesting, though, about BDNF is that a decrease in BDNF can actually improve the impaired learning and memory that chronic stress causes. The question is now exactly how do we decrease BDNF? Okay, well, it's been shown in multiple studies that vitamin E is an effective treatment of impaired learning and memory using animal models. And Biochecki is a study that I want to talk about today because Biochecki measured the effect of vitamin E on learning and memory impairments in juvenile hypothyroidism rats and found one, that vitamin E is an effective treatment of impaired learning and memory, but more importantly, for the proposed study, vitamin E actually acts on BDNF. Okay. In this study, learning and memory is measured using something called the Morris water maze. So imagine a cylindrical tank right here. You fill it up with water, you cut it into quadrants, and there's a platform that you place in the northwest quadrant and you take it in and out. Okay. There are also multiple extra maze cues that are placed around the laboratory room, which provide guidance into locating where that platform is. Rats will go through days of training. During the training days, the platform will be located inside the tank. And the aim of these training days is to get the rats familiar and hopefully they remember where the platform is for the test. During the test stage, the platform will be removed and the time and distance to locate where the platform was originally will be recorded. What Biochecki actually found in these results were that treating hypothyroidism rats with vitamin E improved their learning and memory because the rats that received the vitamin E spent less time and less distance locating the platform. So, however, there are no single study that have actually measured the effect of vitamin E on learning and memory in chronically stressed rats. So, given the evidence that vitamin E acts on BDNF and that chronic stress can impair learning and memory, the research question is now, what is the effect of vitamin E on learning and memory in chronically stressed rats? My hypothesis is that the time and distance will be shorter in chronically stressed rats who receive the vitamin E treatment compared to the rats who don't. So it will be a mixed subjects design. What I'm measuring is time and distance to locate that platform. And the independent variable is the vitamin E. So this right here is taken from the Biochecki study. The vitamin E will be injected into the rats. And again, taken from the Biochecki study, it will be 16 male rats. And in my proposed study, these 16 male rats will be randomly allocated into either the vitamin E group or the control group, okay? And all of these rats will be caged in fours. Here's a visual representation of what the procedure is. And in this first image, you can see it's an induction of chronic stress. So all of these rats will go through this stage, and it's using something called the intruder method. So during week 1, all rats will be remain in the same cage to establish social hierarchy. However, all weeks leading up to week 7, 2 of these rats will be switched randomly from one cage to another to generate that chronic stress. Because these rats have to constantly adapt to these changing environments, that will actually cause that chronic stress. And the intruder method increases plasma levels of corticosterone and blood pressure, which are similar physiological changes to someone who is experiencing chronic stress, and also people with depression and anxiety symptoms. In this second image right here, you can see that the pink rats are the vitamin E rats who receive daily injections of the vitamin E for 7 weeks. And then all of these rats, and also the control rats, don't receive anything at all during those 7 weeks. During week 16, for 6 days, these rats will go through the Morris water maze test. In my proposed study, this tank will be 150 centimeters high, 125 milliliters of water in there, and that platform that I am placing in and out of this tank will be 25 centimeters high. The platform will be placed in the northwest quadrant, and all of these rats will go through 5 days of acquisition training. Okay, so on the first day, they will be given 30 seconds just to explore the maze, and there will be no platform in there. However, during the next few days of training, the platform will be placed in. They will be given 60 seconds to locate the platform. If they don't locate it within the 60 seconds, the experimenter will guide the rat by hand all the way to where the quadrant is, and 60 seconds will be the written down as their time. There will also be a camera that will be placed right above to actually measure the time and distance of the rats, okay? During 24 hours after the very last day of their training will be the test stage. And this test stage is where there will be no platform. The rats will be placed randomly in one of the 3 other quadrants. And then again, they will be given 60 seconds to locate where the platform was originally. And the time and distance to locate where the platform was originally will be measured and recorded and compared against between the vitamin E group and the control group. Now, these are the results that I'm expecting to find. They will be in line with my hypothesis and also in line with the supporting evidence of the Bug-Cheggy study, okay? So my hypothesis, remember, is this sentence right here, that time and distance will be shorter in chronically stressed rats who receive the treatment compared to the rats who don't receive the treatment. And that's exactly what I'm expecting to find, okay? So in this first graph right here, you can see 2 trends. The first trend is that as the rats progress through the different stages of training and that test stage, as they become more familiar with how the apparatus works, they will spend less time each time. But more importantly, when you're comparing against the control group and the vitamin E group, there is a significant difference, okay? The vitamin E group will actually spend less time locating that platform compared to the rats who didn't receive any of the treatment at all. And similar results are actually seen for distance right here. So the rats in the darker bars, you can see the vitamin E group, they will spend less distance locating that platform compared to the rats who didn't receive any of that treatment. And again, that a similar trend is identified where they spend less distance locating the platform as they move through the different stages. So what do these results even mean? Why are they important? Well, remember that this is a novel test, so this has never been done before, which means that these results actually revealed a little bit more about the role of vitamin E on learning and memory in chronically stressed rats, and more importantly on people who these novel results reveal the role of vitamin E on learning and memory in chronically stressed rats, but it also builds upon our current understanding of just vitamin E. What we currently know about vitamin E is, yes, it is an effective treatment, and it does act on BDNF. And this study that I'm looking at actually just builds upon that. In a practical sense, it also provides evidence that vitamin E might be a potential treatment for disorders including generalized anxiety disorder and PTSD, because these two disorders are where individuals experience high levels of stress, and this aligns with findings from the Bug-Cheggy study. So these results actually identify that the time and distance will be shorter when they receive the vitamin E treatment. So thank you so much for listening, and I hope that was interesting.